Hello, students. Welcome to my class. I'm Keerti, and today we are going to discuss about tools and vectors, right? So you already know that these tools and techniques, this all comes under the chapter name biotechnology, right? Let's decode this title first. What is biotechnology, right? Bio means living, that you are already well aware of. And technology means some kind of techniques, some kind of instruments that we are going to explore, right? So, this is all the techniques that we are using to explore the living organism and the processes which they are undergoing, right? So, in the particular chapter, in this chapter consists of various things like various topics, you can say genetic engineering, right? We are working on the molecular level, right? The process which we are going to talk about, or the molecules that we are going to talk about, these are not visible to our naked eye, right? This is something that is happening on a molecular level, right? So this biotechnology is dealing with all kind of techniques using the live organisms or enzymes to produce various useful product that is needed to the human beings. In short, we are going to study all the techniques, all the products, all the processes that is needed to make our life better, right? So this biotechnology is based on two types of principle, or you can say two core techniques. One first one is genetic engineering that I already told you, and second is bioprocess engineering, right? In genetic engineering, you can say the term genetic is involved. It means we are doing something with the genes, and the genes are ultimately made up of what? DNA. You already know the definition of gene is itself says that gene is a segment of DNA, right? Okay, it is having some kind of information. It is coding some kind of protein, right? So genetic engineering, genetic we are going to study about what techniques are there to alter the gene constituent. Okay, whether it is DNA or RNA, right? So genetic engineering deals with the alteration of DNA, and this what will happen, if we are altering the DNA, it means we need to introduce this DNA into some kind of organism so that they can generate the required product that we are going to produce that we need, right? And this bioprocess engineering is actually, is doing it is maintaining the sterile ambience, right? How these products that we want for our uh, for us, how it is going to be managed, right? How all these things are happening in vitro condition, how we can manage the environment, create sterile condition, create particular ambience to grow some kind of cells or the host organism that we are talking about, and how we can generate large quantity of products that we need. For example, the vaccines, the antibiotics, or the particular type of enzymes that we need, right? So, first process is to alter this. After alternation, we need some kind of organism to express this DNA. What will happen? Suppose there is a rose flower, right? It is red, usually the red one. Okay, rose occurs in many colors. Suppose the red one is there, right? But you want this rose flower. I want rose flower, and the scent remains same, everything remains same, but I want purple color, right? How you can do it? This can be done by this genetic engineering itself. It means we have to go up to the level of gene, right? We have to alter a product, a particular gene, a gene that is responsible for the color of that flower. We have to alter that gene, right? What we need a purple color, so we need some information, some kind of external gene that will code purple color. We need to insert that gene in particular, that in that particular area, right? And that gene need to be expressed, right? So for all this process, this molecular process that is going, taking place continuously, we need some kind of tools. It is not easy as we are talking about. It is not like cutting your clothes. You pick up scissor, you want to cut this cloth, you want to make your handkerchief into two, you will cut it into half. This is not an easy process. This is something we are not able to see with our eyes. This is something happening in a very, very, molecular level. We need special gadgets, special tools, right? To, do all this process, a special environmental condition. And after all these, the extraction of the product, whatever the product may be, it depends upon your need, what you want, right? Right? So we have talked about these two processes, prior process engineering and genetic engineering. Now we will go to the major tools, right? What tools we need? These tools include restriction enzymes, some polymerase enzyme, ligases, vectors, and host organism, right? These polymerases, enzyme, and ligases you are aware of in your DNA replication process. There are various DNA synthesizing enzymes, right? These ligases are enzymes which will seal the nick. Whenever there is, a nick present, it will seal that gap, right? In this chapter, we are going to study more about the restriction enzyme. What are these restriction enzymes are doing, right? And the vectors. Okay. So what are these restriction enzymes? See, this restriction enzyme actually act as a molecular scissor. It means it works like a scissor only that we have talked about. But it will not going to cut your clothes, of course. It will going to cut the DNA. The DNA where you want, right? For example, of this is this EcoR1. This EcoR1 restriction enzyme is being extracted from the bacteria E. coli, right? Escherichia coli, right? What will it will do? This will act as scissor. This will cut the DNA. But there is a condition. It will not cut the DNA randomly. It will cut the DNA only where this sequence is present. G double A double T T C. You know the DNA is having these kind of sequences all over the length of the DNA, right? These nucleotides are present. These nitrogen bases are present. So this EcoR1 will act as a molecular scissor. It will go, and wherever the place is where the sequence are present, it will cut the DNA into two. See here, the sequence is present. This will specifically cut the DNA, recognize this DNA, and cut the DNA at this part in both the strands, right? These are anti-parallel strand, right? 5 prime to 3 prime and 5 prime to 3 prime. So it will cut both the DNA, right? So this acts as molecular seizure, right? This EcoR1, I already told you, is derived from Escherichia coli, right? And these restriction enzymes are actually of two types, exonucleases and endonucleases. Exo means outside, endo means inside. Matlab, it means one will cut the DNA within the DNA molecule inside, and what will cut at the end, right? So the naming of restriction enzyme, that EcoR1 that we used earlier, is derived from the organism from which it is derived. See, this E stands for Escherichia. The coli is strain name, right? Right? And this I, this EcoR1 we are speaking, this one depends on which, and R says the strain. What is the strain, right? And I donates the sequence, the what you can say, the time when it was required. If it was first, which it is extracted, so it has written first. If something is extracted in later on, it will written as two, right? Okay. So this is, these are act as molecular seizure, right? This will be used in genetic engineering. Okay. So what kind of sequence that this restriction endonuclease is going to recognize? This sequence is known as, some of these sequences are there. The sequence may be of different types. This is 6 base pair sequence G double A double T C. So when we are reading this in the 5 prime to 3 prime direction, this reads the same. That is why it is known as palindromic sequence. Right? So the DNA is cut. Our next step is, this DNA will be inserted into some kind of plasmid DNA. Right? What is plasmid DNA? Plasmid DNA are extra chromosomal DNA present in bacterium, which divides on their self. Okay. So this DNA will be cut. It will be get inserted into the plasmid DNA. Right? And next, this plasmid DNA will act as a vector. Right? This DNA, this plasmids will act as a molecular vehicle, right? What the process they are doing, they will, pick something from here and will transfer to there. And in this case, whatever they are picking with the linked DNA, and these will transfer the DNA into the host organism, right? So this plasmid will act as vehicle. This plasmid will deliver our interest of DNA, right, into the host organism. Okay. So a new combination of DNA will be formed. That DNA is known as recombinant DNA. Right? So now this DNA is entered into the host organism. And the host organism, now it will make multiple copies. And that particular DNA will also get synthesized multiple amount, right? See this vector. This is the plasmid. This is present in bacteria. Bacteria will have its own genetic material that is known as bacterial DNA, but this is an extra chromosomal DNA, right? So this is the extra part. This will synthesize on its own. It will be not dependent on the bacterial DNA, right? So this plasmid DNA is acting as a vector. What are the characters that is needed for a vector? Some of the most important character that a vector must have is the origin of replication. Why? Because it is not dependent on the chromosomal DNA for the replication process. It must have its own origin, right? It must have some kind of selectable markers, cloning sites, and the ability to integrate with the foreign DNA. Because we are, you know, doing the transfer of our interest of DNA to the host organism, right? So it must have some kind of ability to integrate that foreign DNA into its DNA, right? Okay. So after this, the we have this plasmid DNA. What will happen? See, when our interest of DNA is transferred into the this plasmid DNA, this DNA molecule will be called as what? Recombinant DNA molecule. Because it is having our interest of DNA molecule, right? See what happens is, suppose this is the foreign DNA and this is our vector DNA, the plasmid that we are seeing. We will cut with our molecular scissors, the endonucleases, the EcoR1 that we talked about. This will cut both the DNA, right? So see, some part is being cut here and some part is also cut here, right? Some process will take place. Okay, so this cut part of DNA, what we will do, this DNA we will get inserted into our plasmid DNA, right? So this is the foreign DNA that the plasmid is carrying. This recombinant DNA molecule will now transfer into a host organism. Why host? Because the host is accepting this DNA, right? And after that, when this host will divide into multiple numbers, say in case if we are saying since this plasmid is coming from bacteria, so the host organism must be bacteria. So when this bacteria will divide n number of times, this our interest of DNA will also get duplicated, replicated in n number of times, right? Okay. So after this identification, this process, we have bacteria, right? In which there is their own DNA and they have extra chromosomal DNA with our interest of DNA, right? See, but the selectable markers are nothing. In lame terms, I want to give an idea that these markers are used to find out whether the particular cell is having our DNA or not, right? Some of these markers are, for example, the antibiotic resistance. Right, right. Suppose this bacteria now are taken into a Petri plate, right? This have ampicillin and this has not. This also has ampicillin, right? But this DNA is not carrying our gene of interest. This is simple plasmid DNA. What will happen? This cell, which is not having the ampicillin resistance, right? This will not grow, right? And the other slide, the other, plate, that is having the ampicillin and it is having our foreign DNA, it will go. So we will deal in later how they are selected. In this session, I'm going to just give an idea that these are acting as a molecular well. This we will deal what is happening, how ampicillin is working, why are using ampicillin. Okay, when we are using tetracycline, we will deal because it will get too much in this particular you know session. You won't be able to make yourself clear what I'm trying to say, right? So the basic idea is, let me you know just wrap up the simple process in a very simple way. Suppose this is the DNA, right? And this is our vector DNA. This will what act as a transport mechanism. Suppose this is our cycle, right? What we will do, we will use endonuclease, cut will DNA, cut this DNA. This will generate fragment, right? This is our gene of interest, and this vector DNA is also cut. Now we will fuse this both, and this will have our gene of interest, right? This is plasmid DNA. Okay. Okay. Now this plasmid DNA we will transfer into the bacteria. Okay. And this bacteria will now, what will happen? This will divide and create various bacteria, right? Okay. So this is the cut short process of recombinant DNA technology. Recombinant DNA. This is what? This is a recombinant DNA, right? We have excluded some part of this plasmid DNA and have added our interest of gene into it, right? So this is what a recombinant DNA. Recombination is taking place and another new molecule is formed. So this is all about this recombinant DNA technology. We have tools. What were the tools? Of course, the nucleases that we have talked about. This joining, when this joining is happening, some enzyme like ligases should be there, right? To join this nick, right? We have polymerases which will synthesize the DNA, right? So all these tools will help in creating a recombinant DNA molecule. Okay, hope I'm clear. This is a very cut short video. Don't make yourself in a hustle situation, right? This we will go in deep so that you can have a stepwise idea. This is just an overview of how vectors are working, right? How these vectors are transferring our gene of interest into the host cells. There's host cell, maybe any organism. When we are talking about this particular session, I have said that it is bacteria. Okay, so we will deal in a very deep manner. Hope till here you are okay with this. I am going to I'm going to teach in deep. You don't have to worry. Even if regarding this session you have any doubt, you can contact me through my Urban Pro profile. When we are going to study very much deep into the chapter, I'm going to tell you each and every part in a bit detailed way, right? This is just an overview. Take it as an overview. Just grab whatever points your brain is going to catch in this first session. For example, the eco R1, what is exonucleases, right? What is vector? What is recombinant DNA? You must have, you know, somewhat idea what is happening with all these processes, right? So hope I'm clear. We will do, of course, we will study deep, very deep. Okay. Till then, take care, take revising, go through your NCRT. That is our basis, right? So you must have an idea. Go through it. And again, I'll say if you have any doubt, you can ping me, you can contact me through my Urban Pro profile. Okay. Thank you. Thank you for attending this session. Thank you. Take care.